Hey guys, we're here with chapter 4, which is the biology of the cell. This is going to be an awful lot of review of concepts that you learned in Bio 101. It's going to be a lot of cell organelles and cell membranes and what they're made of and what they do, and sort of how that all plays into human anatomy, which will be maybe the new part for you. But let's go ahead and dive in. We'll see that we do have a lot more objectives for this section than we had in our previous section. So we're not going to spend a lot of time here, but basically it's knowing the parts and pieces of cells and what they do. So what is a cell? A cell is the basic unit of life. It's the smallest living thing. If it is less organized and smaller than a cell, it is not living. So something like a virus, not alive. Something like an atom, not alive. But once we get this organization and we get a cell, we now have a living thing. We divide our cells into three main parts. The first is our cytoplasm, and that's just this cytosol, this goopy jelly stuff, and any sort of stuff that is in it, like ions and molecules and that kind of thing. And then we've got our nucleus. That's our control place in our cell. And then we've got this plasma membrane. So let's first start with our cytoplasm. Again, that's just the term for everything that is between the plasma membrane and the nuclear membrane. So it's going to include that goopy jelly stuff and all of the atoms and molecules that are in it, as well as our organelles. The cytosol, that is the fluid, the sticky, goopy jelly stuff that is made again of organic and inorganic molecules. All right, so let's dive into the different cell organelles. First, we've got ribosomes. Ribosome is the site of protein synthesis. So you can sort of think of them like the protein manufacturing plants. And we know that we're going to need messenger RNA and transfer RNA as part of this process of making proteins. We have the endoplasmic reticulum. That's going to be this network of these different sort of membrane channels. And it's going to be really this continuous bending, folding of these channels that go from the nuclear envelope out into the rest of the cell. The endoplasmic reticulum comes in sort of two flavors. We have the rough ER, which has ribosomes attached to the membranes. The rough ER is responsible for modifying or processing proteins that are made by the ribosomes. So you can sort of think of them as like a production plant. The smooth ER does not have any ribosomes that are attached to it. It's just smooth membranes. And its job is to make lipids. It also will detoxify, get rid of any toxic compounds that get made or are found in the cell. And it stores calcium. The Golgi apparatus, that's another sort of folded up membrane, or you can think of it like a little stack of pancakes. It is going to get vesicles, these little bubbled off pieces of membrane from the endoplasmic reticulum. It's going to package them up and ship them to other areas of the cell. Now they can ship it out of the cell in secretion, or they'll ship it to a place in the cell that needs those products. So the Golgi apparatus is kind of like the UPS or shipping facility of the cell. We have lysosomes. Lysosomes are going to contain enzymes that can break down molecules. They sort of have three functions that we need to look at. They will do phagocytosis, which we call cell eating. They'll actually make a food vacuole. So if there's any particle that needs to be digested, that lysosome will come and sort of engulf the particle that's happening in this little first picture here, and then make a food vacuole to digest those product or to digest those pieces and get products that we need. It can break down or digest non-working organelles. So just as you know, you think of like a human body aging and parts and pieces becoming less useful. It happens even at the cellular level as the cell ages. So we need to get rid of the non-working organelles so that we can get working organelles or replaced and repaired, or if the cell is old, that it would then die. And we call this process of breaking down non-functioning organelles autophagy. And that process of the cell being programmed to die, whether there is an error or it's old, we call that apoptosis. And that is programmed cell death. Peroxisomes also exist as an organelle in certain cells. They have oxygen-based enzymes that are going to detoxify and break down fatty acids. The mitochondria, you probably remember from other bio classes, the powerhouse of the cell. It's where we're producing ATP that is used as the energy for the cell. It's important in cellular respiration, which we talked about in the previous chapter, where we take glucose and oxygen and we make ATP, carbon dioxide, and water. The outside membrane of the mitochondria is smooth. The inside membrane has many, many folds, and we call that the cristae. And that, of course, is where we are having this cellular respiration take place. Animal cells have centrioles. Their job is going to be to organize mitotic spindles in mitosis. And really, they look like little pieces of Twizzlers or pasta. And they're kind of just hanging out until it's time for the cell to divide. And then they will go to opposite sides of the cell and sort of organize the framework to move the parts and pieces around the cell as it divides. There's some surface structures we should look at. Cilia are just short hair-like structures, and their job is to move substances over tissues. If you remember, tissues are just a bunch of cells that work together. So these cilia will move, and it will move things across those tissues. A great example is the trachea. You've got these little cilia on the cells to help, move air and get rid of tiny particles out of that air. We also have flagella. Flagella are these long whip-like structures, sort of like a tail, if you will, and that's used for motion, locomotion. And then there is microvilli. These are like little protrusions or extensions of the plasma membrane. It is there to increase the surface area, and that way we can absorb nutrients, minerals, vitamins from the area around it. And you'll find these, for example, in your intestine. Of course, we have the nucleus of the cell. The nucleus is going to contain all of our genetic material and genetic information for the cell, and that is in the form of DNA. If we look at the nucleus, we need to break it down into more components. The first thing that we have is the nuclear envelope. That is what actually encloses and protects the DNA. It is a double membrane and it is a lipid bilayer. Next, we have the nuclear pores. These are like little holes or pores in that nuclear envelope that are going to allow the entry and exit of molecules from the nucleus. When we look at the DNA itself, we find it in sort of two forms. We can have chromatin. That is when a cell is not dividing. The DNA is just in these little strands. And then there are proteins also present that are called histones. Then we can have chromosomes. Chromosomes happen when the cell is about to divide. The chromatin gets tightly condensed or coiled, and it will actually wrap around those histones. You can sort of think of it like chromatin being your headphones that have cords that are just thrown in your backpack. And then the chromosomes are when you take those headphone cords and wrap them up nicely so that they don't get tangled. In that nucleus, we call the nucleolus. Inside of the nucleus, the part where we have RNA, and that is where we'll actually make our RNA, which we need again in order to make proteins. So, how does this making of proteins work? Well, basically, it is directed by the DNA. We have first transcription, where we're going to make a copy of the gene that is in the DNA in the nucleus. So, we are taking the DNA and we are making a messenger RNA. Then we have translation, where that messenger RNA is going to head to a ribosome in the cytosol, and we are going to then use transfer RNA and make a protein. We also have our plasma membrane, which is what makes our cell a cell. It is a phospholipid bilayer. It is going to regulate what goes in and out of a cell. You can think of it like a gatekeeper. And it is also where we have some other um, things embedded, like proteins that maybe are used as channels or are integral into the membrane. Or we can have steroids like cholesterol in that membrane. So while most of it is phospholipids, we do have other structures there as well that help with that regulating of material in and out of the cell. Some different parts and pieces that are there, we have glycolipids. Those are going to be phospholipids with a carbohydrate attached. Think glyco like glycogen, carbohydrate, and lipids, those fats, lipids. The word tells you exactly what it is. They are going to be located on the outside of the cell, and they form what we call a glycocalyx, which is going to be sort of this network the cell uses to identify itself versus something that is not itself. And it helps our cells stick together. It's very important that our cells can recognize, like, hey, I'm part of this body, I belong here, or things that are foreign invaders that don't belong, a non-self cell. So these glycolipids, as you can see in this top picture, kind of are like a barcode that an immune system cell, a white blood cell, can identify that what is good and should stay and what is an invader that could cause harm. And then this is just sort of a zoomed in version of that. You've got your glycolipid attached on this membrane. All right, let's look at the proteins that are embedded as well. You can have a channel protein that is going to allow things in and out of the membrane, usually small things that could easily move. You've got a carrier protein that is going to sort of change shape, and that might require energy in order to move the substance in or out of the cell. You can have pumps that do require energy and are usually working against a gradient to move things out into a matrix area or outside of the cell. You can have a surface cell receptor, so we need to have something bind to this protein in order for it to do something. Of course, we can have enzymes that make chemical reactions happen. We can have anchoring sites to help get junctions between cells. And then, of course, we can have cell adhesion proteins, which again will help stick cells together. Um, and those are some different kinds that we have. Let's kind of switch gears now that we know about our cell membrane to talk about those things that are moving in and out of our cell. So we know that that cell wall or cell membrane is there as a way to gatekeep what can move in and out. But obviously, we need things to be moving in and out of our cells. So, how does this all happen? Well, there are passive processes that do not require energy. We're going to specifically look at diffusion, osmosis, and facilitated diffusion. And then there are active processes that do require ATP or energy in order to work. We're going to look more at active transport, vesicles, and sort of how these work as well. In order to do that, we need some special vocabulary. So let's start here. First, we need to know what is a concentration gradient. That is just the difference between the concentration of a solute in two areas or two spaces. You can sort of think of it as the analogy of like an air freshener. If you stand in one corner of your room and you spray Febreze or a perfume or an air freshener of your choice, it is highly concentrated right where you are standing, where you just sprayed it. However, given time, those particles will move and be more equally distributed around the room. And then the concentration will be sort of lower than where it started, right? We've lowered the concentration gradient because we've spread those molecules out. Same thing here. We've got a high concentration of this blue molecule. If it moves down the concentration gradient, it spreads out. If it's going against the concentration gradient, well, then it will move towards where there is already more of it. When we move things down a concentration gradient from where there is a lot of it to where there is less of it, we really end up calling that diffusion. And it's passive. It doesn't require energy. The molecules just already want to spread out. And there is some physics in there with Brownian motion if you want to look that up more. But essentially, those particles want to get away from each other. If we have to move it back from that low concentration into a high concentration, that is going to require energy to do that. And those are our active processes. Something very important to note, we can reach equilibrium when the concentration is the same. Now, the movement of these molecules isn't going to stop. It is just that the rate that they are moving up and down, the concentration gradient will be about equal. So we end up with the same concentration. So again, active transport, that is going to be going from this low concentration into the high concentration. Active transport requires ATP. You can think of AT in active transport as requiring ATP. If we've got diffusion, we're taking molecules from a high concentration, moving to a low concentration, doesn't need energy. We've got D in diffusion, D in doesn't need energy. And then osmosis is just a special word for moving water molecules. And so moving it from this high concentration of water molecules to a low concentration of water molecules is osmosis. So let's first talk about passive transport. Diffusion and osmosis are the processes we're going to look at here. Diffusion is just that movement from greater concentration to lesser concentration. And that is going to keep happening until we reach this equilibrium. And again, equilibrium doesn't mean the particles stop moving. It's just that they are uniform throughout. We can have a process called facilitated diffusion, where there are molecules that are kind of too big to sneak through that membrane. So there will be a type of protein embedded in the plasma membrane that will move these substances down the gradient from where there is a lot to where there is less. So it doesn't need energy to do that. It just needs a way to get through the membrane. Osmosis is moving water through a selectively permeable membrane, like a cell membrane. The membrane will let water through, but large solutes or charged particles in solutes, they can't get through. They're stuck. So the water is what will move to sort of compensate for the solute particles not being able to move. And that will make a difference in water concentration on either side of the membrane. When we look at osmosis, we're mostly talking about tonicity. And tonicity is the ability of a solution to cause a cell to either gain or lose water, which affects, of course, the pressure of a cell. And it's going to depend on the concentration of solutes inside and outside of the membrane. So these are relative to one another. You cannot really just say like, oh yes, that is an isotonic thing. It needs to be isotonic to the solution it is in. That's what I mean by relative. And there are three main types: isotonic, hypotonic, or hypertonic. An isotonic solution has the same concentration of solutes inside the cell membrane as outside the cell membrane. So it's like putting a cell into this solution here, and we have the same amount of stuff. That means things will move in and out of the cell at the same rate, and nothing happens to our cell. Its pressure stays the same. It is just living its life normally. A great example is you may have to get an IV drip, and it would be of this salt solution, sodium chloride solution. And basically that is just keeping your body fluids. It is the same solute concentration as your cells, so therefore we can use it to keep the blood fluid at a homeostasis at a good level, basically. Then you can have something called a hypotonic solution. There is a lower concentration of solutes outside than inside. And we can think of this as like putting a red blood cell in just pure water. There's more stuff that is inside the cell than outside the cell. Because of that, water moves down its concentration gradient from outside the cell to inside the cell. It increases the pressure inside of the cell and causes lysis, causes it to break. Specifically in red blood cells, we call it hemolysis. And that's what's happening here. We've got these red blood cells in pure water, and because there's more stuff, more proteins and sugars and salts inside of this cell, the water rushes in, it swells, and then it bursts. One way that I like to kind of try to think of this is hypo kind of sounds like hippo, and hippos live in water and they get big and fat. So if that helps you, then great. Remember that things put into hypotonic solutions, they will get big and fat, they will swell, and eventually they will lyse. So here is another example that we have. We've got a solution outside of the cell that has 10% salt and 90% water. Inside of the cell, we've got 20% salt and 80% water. There's more stuff, more solute in this cell. So the water will go inside of the cell. That will cause the cell to swell up, and eventually it could lyse or burst. Hypertonic solution is the opposite. There's a higher concentration of that solute, the higher concentration of solution outside of the cell than inside of the cell. There's more stuff in the solution than in the cell. An example would be like putting a red blood cell in a 3% salt solution. Because of that, water is going to move down the concentration gradient, and it's going to go from inside of the cell to outside of the cell. You can think of it as trying to dilute that higher concentration of stuff. When this happens, we decrease the volume, we decrease the pressure of the cell will shrink, and we call that crenation. So here's what we've got going on. We've got this environment where we've got a 15% salt solution and 85% water. Inside the cell is only 5%. And then we've got 95% water. Because there is more stuff on the outside, right? 15 is bigger than 5. The water that is inside this cell will move out. So what is osmotic pressure? What are we talking about here? Osmotic pressure is just the pressure that is exerted by the movement of water across this semi-permeable membrane. And it's due to the solution concentration differences. If there is a steeper gradient, the water is going to move by osmosis, and there will be this change to a greater osmotic pressure. Hydrostatic pressure is the pressure that's exerted by a fluid on the inside wall of its container. So that is just the pressure that is going on the wall of the tube or in our bodies, in our blood vessels, that pressure that we can feel and we can measure. So here's what it would look like to put red blood cells, this comes from your book, in these 3 different types of solution. If we're in an isotonic solution, there's no net movement of water. It's moving in and out of the cell at the same rate, so we have a normal red blood cell. It's functioning how it should. If we get a solution that is hypotonic, remember those hungry hippos, it's going to bring water into the cell. The cells will swell, and that's okay if it's a little bit, but if it keeps having this hypotonic solution around these cells, they can burst, and that can be hemolysis. Opposite is this hypertonic solution. And I forgot my little device to remember that. I think of hyper, like hyper children that you need to take outside so they can run around. Same thing. Here with our water leaving, going outside of the cell. because the concentration is higher outside of the cell with these solutes. So we're going to try to dilute it. Water leaves, or like taking a hyperkit outside. So here's some basic practice for you to look at. And this is something you do in Bio 101, but it may have been a while or it may not have ever really made sense. So of course, please take some time here. Can you identify, are these hypertonic, hypotonic, isotonic? And then I have a little answer key that gets posted for you. But take some time now, pause the video, and go through. Can you identify these solutions? All right, let's look at active transport, the parts that require energy in order to happen. So first, active transport is just moving molecules against the concentration gradient. If there's a low concentration and we are moving it to a higher concentration, this will require ATP to do this. And we have something called a protein pump that we can have to do this type of transport. The most important one that we look at is the sodium-potassium pump. It is going to exchange two ions. In order for this pump to work, 3 sodium ions will go into it and 2 potassium ions will go into it, and then they are moved across the membrane. Of course, this does require energy to do. So we want to bring 3 sodium into the cell. We want to release 2 potassium. How does that work? 2 potassium from inside the cell go into this protein pump. 3 sodium from outside of the cell go into this pump. When we add energy, we're able to switch them across our membrane. So we get our sodium in, and we will release our potassium. There's also something called vesicle transport. Also, you'll read it being called bulk transport. And this is where we're going to transport large substances across the plasma membrane by making a vesicle. And again, a vesicle is just this membrane sac that gets filled with materials. There's 2 major types. We have exocytosis, pushing things out of the cell, or endocytosis, bringing things in. And our little graphic here shows you how this can work. You will have some sort of structure in the membrane that will sort of act to hold on to molecules, and then they can be brought in, and the membrane will shrink around it. With exocytosis, we are taking things outside of the cell, and that word exocytosis breaks down to meaning exo, outside, cyto, cell, so outside of the cell. And again, that's going to require ATP. How that works is you have your vesicle filled with the stuff that you want to release. It is going to move to the membrane, attach to the membrane, and then it kind of like unfolds or unzips, and anything that was inside of that vesicle is now on the outside of the cell. Endocytosis, endo inside, so we're bringing something in. And then we can have kind of 3 types of that. We can have phagocytosis, which is considered cell eating. So again, that membrane will sort of pinch in and scoop up these particles that the food or that the cell wants to sort of eat. Penocytosis is cell drinking. So that's when those molecules are sort of dissolved in a liquid like water. Or we can have receptor-mediated endocytosis, where there are those receptors on the membrane that will only form a vesicle when we have enough of a substance. So again, phagocytosis, we're going to have these false feet or pseudopods reach out and engulf this large particle. There's only a few kinds of cells in the human body that can do this. There are plenty of protists that do it in like the animal world. But in humans, we're really kind of limiting this to white blood cells engulfing microbes in order to stop an infection. Pinocytosis is cell drinking. These are particles that are dissolved, usually in water. It makes this little pit or little, sort of pore here that will pull in these dissolved particles and then bring them into the cell. This is much more common in cells to be able to do this type of endocytosis. And then, of course, the cell-mediated, it's a type of pinocytosis. It's just requiring a receptor molecule to sort of hold on to these solutes, and then it will only make that vesicle when we have, enough of those solutes present to be pulled into the cell. Okay, switching gears almost entirely, but dealing with that sodium potassium pump, moving ions, let's talk about resting membrane potential. So, plasma membrane is there to establish, or I guess can establish, this electrochemical gradient. And that difference in charge from inside and outside of that cell is essential for muscle and nerve function to happen. So, what are we even talking about when we talk about this? We've got an electrical charge difference, and that's our membrane potential. The resting potential is when the cell is at rest. It's not actually firing, doing work. It is just sitting there. And it happens because of this unequal distribution of ions because of those sodium and potassium pumps. Okay, so we've got a high concentration of sodium outside. We've got a high concentration of potassium inside. But there's a difference. So inside's got a negative charge, outside has a positive charge. So what is happening is, of course, we have these pumps moving our sodium, and we have them bringing in our potassium at this sort of unequal rate. And so when it's resting, we have what we call a 50 to 100 millivolt potential. That's just measuring the electric current because of these ions. And then though, when these muscles or these nerves sort of fire, they need to work, we change this potential and we could measure it as, volts, and we get this negative 70 millivolts. And essentially what happens is once we fire that neuron or that muscle cell, we have to wait a little bit of time to reestablish this resting potential before it can work again. It can't just constantly be firing. We have to reestablish that gradient. And for now, that's really all you need to know. We can dive deeper in, and we will dive deeper in when we get to nerves and when we get to muscles. But just for now, knowing that there is a difference across the membrane when that muscle or nerve fires, we have to wait a short amount of time, a recovery period, before it can work again. Okay, so let's talk about cell division. You definitely cover this in lab, but you are responsible here in lecture as well. So cell division is in two flavors or two different parts. There's mitosis, which is somatic or body cells, anything that is not a sex cell. One cell divides and then makes two cells, and it's an identical copy. And of course, we need this for development, tissue growth, replacing old cells, and repairing tissue. Then there is meiosis, and that is the division that makes sex cells. It is what is going to eventually give us sperm and oocytes, or a lot of times you call it an egg. When we talk about mitosis, and to some extent meiosis, we need to talk about a cell cycle. And that is just the steps that are in the replication of somatic cells. It is how we are going to divide a cell into two identical daughter cells. And it's in two major parts. There's interphase that has a growth 1 phase, a synthesis phase, and a growth 2 phase. And then there is the mitotic or M phase, where you have your PMAT, your prophase, metaphase, anaphase, telophase, and then cytokinesis. So in interphase, what is happening is this is the time that the cell has to prepare to divide. The DNA starts as this loosely coiled chromatin, right? Sort of like those headphones just tossed in a backpack. And again, there are 3 phases, the G1 or growth 1, S or synthesis, and G2 or growth 2. In the G1 phase, we need to grow and make new organelles. If we are dividing and splitting up, we want to have 2 of everything so that we can have the same exact cells when we finish. So we need to make double the amount of organelles. It is also where the structures for DNA replication are formed, right? Because we need to make a copy of our DNA before we split the cell. Then we've got our S phase. That is where we are synthesizing that copy of DNA. So this is where we are doing the copying. G phase is where we just get ready to do mitosis. It's sort of a growth and a rest phase before we start actually dividing. In mitosis, we, of course, are going to have that after interphase is done, after we get G2. It is going to be where we make the 2 new cells. And of course, with mitosis, that's really what we call the division of the nucleus. We sort of lump it all together to mean cell division, but really it's how we split up that copies of DNA. And then cytokinesis is how we split up the cytoplasm. There's 4 steps. We've got prophase, metaphase, anaphase, telophase, and I like to just remember PMAT. I watched a video, and I have that video on the D2L for you, but that's where it came from, and it just stuck. If you've got a better phrase to help you remember, please share. I'm happy to learn more. All right, with prophase, the chromatin first is going to condense to make chromosomes. The nucleolus will break down. The spindle fibers will form. Centrioles will move to opposite sides of the cell. And we're going to lose our nuclear envelope. So we're sort of opening up the nucleus because we are going to be separating our DNA to the 2 ends of the cell in the next stages. Metaphase is where our sister chromatids are going to line up in the middle of the cell. I like to think of M for metaphase, M for middle. Anaphase is where the sister chromatids separate and move away from the middle to go to the poles. And I like to think of A for anaphase, A for away. and they also sort of look like little sideways A's as we're moving them apart. Telophase, that is sort of the opposite process of prophase. And it is where the chromosomes are going to relax and become chromatin. Our nucleolus is going to reform. Our nuclear envelopes reform. Spindle fibers break down. The centrioles move back to the nucleus because they're not going to be on opposite sides of the cell. So it's sort of this like finishing up end process. And then cytokinesis, that is going to be dividing the cytoplasm between two newly formed cells. They might overlap with anaphase and telophase a little bit. We'd like in biology to set things as like separate boxes as we learn. But when you look at this process under a microscope, it's not as smooth as, what we would call, PMAT here. It's sort of overlapping a little bit. How is this working? There are these ring of microfilament proteins that are really just pinching and squishing the cells so that we get two separate cells. And that pinching in area, we call it cleavage furrow. And that is where we're going to get two daughter cells. And after cytokinesis is done and we've, pinched off these two membranes and we've separated the cytoplasm and we now have two cells, cell division is complete and we go back into interphase. So with this, the last part of your text kind of talks about cell aging and cell death. Because, we just talked about, reproduction of cells, really making new. So what goes on here? Well, aging is a normal process. As you go from that cell that just divided and is now back in its interphase, doing its cell functions. Parts and pieces will wear out, and that is aging. It's a normal thing. And scientists are still really digging into the processes of aging because not everything is well understood. We have sort of a general idea about cell aging, especially with DNA and having parts of DNA degrade. And those parts we call them the telomeres. That's really not important. We just don't understand all of it. Some examples of things we see in a normal aging process, though: mitochondria begin to fail. There is change in the chromatin and chromosomes, and just overall not working as it once had. Cells die by two main mechanisms. They either get killed by harmful agents or they are induced to commit suicide, or that programmed cell death that we call apoptosis. So again, apoptosis is this very ordered, well-regulated steps. It's run by a series of enzymes that are going to degrade and remove cellular parts. And kind of the order that this goes is we're going to start by destroying DNA polymerase. So we cannot make new DNA. If we don't make new DNA, we don't do mitosis. We're going to break down the existing DNA into small fragments. We'll start to digest the cytoskeleton. And if we don't have the cytoskeleton as the support, our cell is really going to start to fall apart. And we see that when we have these formation of small blebs or bubbles on the plasma membrane, because we no longer have our cytoskeleton kind of holding things as they should be. We get this squishing or condensation of the cytosol and destroying the organelles. And finally, digestive enzymes are released and we essentially break down the cell parts. And this happens in our aging old cells. It can happen during development. So, if we look at embryology, we at one point have these little paw paddle hands that we then, basically have apoptosis of the cells to make our fingers and our toes, and we lose our tail. So we need some of this for that purpose. And we have apoptosis also as a way to remove harmful cells, cells that have been infected by viruses or have mutated DNA. So how does this play into cancer? Fundamentally, at the very basis, cancer is unregulated cell growth. So that there are errors in that mitosis, in that cell cycle, and errors in the process of apoptosis. Of course, we all recognize now that there is still so much to learn about the mechanisms in the cell cycle, about the mechanisms of apoptosis, and how that relates to cancer and tumor growth, and how we can, sort of hack these or utilize these as potential treatment. So, your book talks very little about cancer in this stage. Every time we go through a body system, we do recognize the aging and cancer portion of it. So if this is something very interesting to you and this is just not enough info for you yet, don't worry, it's coming. Your book is just setting the stage that we do have a lot to learn, and there are advances happening constantly in this field. So, of course, if you have any questions, please feel free to let me know. I am happy to speak with you further. There are lots of great resources on the D2L for you about these different processes, about the cell organelles, about cancer and mitosis. So, feel free to dive into those. And of course, thanks for watching and hope you have a great rest of your day.